molecule that contains all of the information that our cells need in order to survive. It's a linear molecule that's packed really tightly inside our cells in a way that resembles tangled strings. And so when a cell needs to divide, it has to find a way to take this tangled mess of DNA, make a copy of it, and then separate these two copies into what will become two new cells. So if we were thinking about how we would do this with string, a way to organize string is to take each strand and wrap it around a spool. This allows you to separate each strand and move each one individually. Cells use a mechanism really similar to this to organize their DNA during cell division. They take each strand of DNA and wrap it up around proteins in order to form structures called chromosomes. Now, each human cell contains 23 pairs of chromosomes, so 46 total, and our chromosomes are shown here. And they can vary in size, and then you might recognize the sex chromosomes, which individuals will have either XX or XY. Now, these 23 chromosomes contain, each chromosome contains a different subset of the information that our cells need in order to survive. So it's really important that each cell contains the proper number of chromosomes and also that there is no damage to DNA. If either of these things go wrong, this can lead to two groups of issues in humans. First is developmental disorders, if there are issues in cell division during development. An example of this is Down syndrome. This is also known as trisomy 21, and this is because these individuals contain an extra copy of their 21st chromosome, showing you that even just one extra chromosome can actually have a large impact on an entire organism. And a second time where cell division is really important is during cancer development. Cancer is a group of diseases where the cell division machinery is mistakenly reactivated, usually later on in life. This leads to cells dividing uncontrollably, which gives tumors the ability to grow and spread throughout someone's body. So when we look at the chromosomes in a cancer cell, this is a specific example from a leukemia patient. We once again see issues with the number of chromosomes. For example, chromosome here has copies, but we also see damage to chromosomes where parts can be missing or completely fragmented. So these are just two examples, but it's extremely important for human cells to have the correct number of chromosomes and for their DNA to be kept safe and not be damaged. And so when a cell is not dividing, they have a really good system in place to protect their DNA. Like I told you, DNA is this tangled mess, but it's surrounded in the cell by what's called a nuclear envelope. And just like you put a letter in an envelope to protect it when it goes through the mail, the nuclear envelope protects DNA from the other contents of the cell and prevents it from being damaged. So DNA inside the nuclear envelope is called the nucleus. And this is kind of the main structure in the cell through which information is sent out and different proteins are made. And so in my lab, we use a lot of microscopy. And shown here is an image of a human cell. You can't see the outline of the cell itself, but you can see we are staining DNA in blue, and you can see it's very tightly packed inside the nucleus. And the nuclear envelope is shown here in green, and it forms this nice wall around the nucleus that will protect DNA. But when cells need to divide, they have to find a way to form chromosomes and separate two copies of DNA while still protecting DNA during this process. So they have this list of steps that the cell will go through in order to regulate this process and prevent errors from occurring. So when a cell needs to divide, the first step is that chromosomes will form, where each strand of DNA is wrapped up around proteins, and this protects DNA from the contents of the cell. Because also at this step, the nucleus will break down, where the nuclear envelope is disassembled and the chromosomes are released into the cell. Next, these chromosomes will be lined up in the center of the cell and will be attached to structures that work like molecular ropes. And these ropes will attach at a specific site on each chromosome, and two ropes will bind each side coming from each pole. And then once all of these chromosomes are properly aligned and attached, these ropes will do their job and pull the chromosomes apart towards what will become each new cell. Once these chromosomes are separated, the nuclear envelope will then reform around each copy of DNA. And then once this nuclear envelope is reformed, the chromosomes can unwrap, allowing the cell to access information and proceed on with its normal functions. And so, like I said, this is a very controlled process in cells, but it's also very easy for errors to occur. And so there's a couple different ways in which cell division can go wrong. One possibility is that there is a gain or loss of chromosomes. And this is what happens with Down syndrome and trisomy 21, where one cell receives too many chromosomes and one too few. Another possibility is that the two cells fail to separate, leading to one large cell with two nuclei that will eventually die, which has negative effects on an individual if their cells are dying. And then one other possibility is that after cell division, the chromosomes can fail to form one nucleus, and actually two nuclei can form inside a cell. One is called the micronucleus. And so this is the form of cell division error that I study in my lab, and I want to go through in more detail exactly how a micronucleus forms in a cell. So as I mentioned, an essential step in cell division is that all chromosomes are aligned in the center of the cell and attached by these ropes. But sometimes it's possible for one chromosome to fail to attach in time. So if it's delayed in this process, once chromosomes are pulled apart, this can lead to chromosomes lagging and kind of being separated from the main chromosome mass. We can see this using microscopy, where here I'm showing you DNA in a human cell. And you can see the two large groups of chromosomes being pulled apart. And then there are these two smaller chromosomes kind of stuck in the middle, and they're isolated from the main groups. And so if those chromosomes stay isolated, they can actually form their own nucleus, so that once the chromosomes unwrap, this cell on the left here now has one primary nucleus containing the majority of chromosomes, and then one smaller micronucleus that contains one or a few chromosomes that were isolated because they were lagging behind. And so that's all a micronucleus is. It's really just a smaller nucleus. It's made up of DNA surrounded by a nuclear envelope. Once again, this is an image showing you a micronucleus inside a human cell. And we commonly see micronuclei in cancer cells. This is most likely due to the fact that cancer cells are dividing very quickly and making a lot of errors, leading to micronuclei. And so at first it seems like while forming a micronucleus is not ideal, it might not be that bad for a cell. All the DNA is protected inside a nuclear envelope at the end of the day, so it shouldn't really be that detrimental. But it turns out that micronuclei are very unstable and they frequently rupture. And what I mean by rupture is shown here. There is a gap in the nuclear envelope where a hole can form and expose DNA to the rest of the cell. And so we don't know actually what causes this. People are still trying to understand what is causing micronuclei to rupture, but we do know that this rupture has a lot of negative effects on a cell and on an individual. First, exposure of the DNA to the cytoplasm leads to large amounts of DNA damage. DNA is supposed to be protected when it's unwrapped and not in its tightly bound chromosome form. And so chromosomes can be, this DNA can be fragmented, and it can damage the information contained on the chromosomes in the micronucleus. Another bad thing that happens is that this can lead to an antiviral response. So cells are not used to seeing DNA outside of the nucleus. And in fact, the only other time this really happens is when a cell is being infected by a virus. So if a micronucleus ruptures and exposes DNA to the rest of the cell, this cell can actually will respond like it's being infected. And if this happens in a cancer patient, it can lead to unnecessary inflammation and immune response in that patient, which can contribute to cancer progression. And so both of these responses to micronucleus rupture can contribute to cancer progression. They not only occur in cancer cells from errors, but they can lead to more errors in the future. And so understanding exactly what micronuclei are doing in cancer cells and how they form is really important for understanding how we can treat cancer patients. And so despite our knowledge about some of the negative effects of micronucleus rupture, there's still a large gap in our understanding of how these small structures form. And that is that we don't know which chromosomes are contained inside micronuclei. And so, like I told you, we have 23 different chromosomes in our cells, but we don't know whether specific chromosomes are more likely to form micronuclei or if this is a completely random process. And so we believe that this is not a random process because it turns out that even though all 23 chromosomes are made up of DNA, they're actually very different from each other. And they have a lot of different features that could impact their response to errors in cell division. So in order to understand these differences between chromosomes, I want you to think of chromosomes like people. So if we look at a group of people, we know that they're all human beings, but they all have a lot of differences that make them unique. Some differences we can see with our eyes, such as differences in height, hair color, or style. These are easily identifiable. But if you dig a little deeper, there's also differences between people that are not visible. And an example of this is people's personalities. Some people might be more extroverted, while others might be more quiet and introverted. So really, each person is unique because of a combination of both visible and invisible features. And so these same types of features are seen on chromosomes. Here I am showing you all 23 chromosomes in the human cell, but just one of each pair. And they're drawn to scale. So first, to go through visible features that we know differ between chromosomes, what I think is easy to see from this diagram is that they really differ in size. Chromosomes were actually named or numbered based on their size. So from chromosome 1 to 22, there's kind of a gradual decline in size of each chromosome. Another difference between chromosomes that we can see is, like I told you during cell division, chromosomes are attached to molecular ropes at a specific site. And so we actually know where this site is on each chromosome. And it's shown here in gray. And so the specific attachment site can vary in where along the length of the chromosome it's located, and also in its overall size. And the size of this attachment site doesn't necessarily scale with overall size, where chromosome 18 has the largest attachment site, but is actually small on overall size. And so these are just two examples of features we can actually see when we extract from. They can also differ in ways that aren't necessarily visible. One way is in the number of genes contained on each chromosome. So as I mentioned, each chromosome contains a different subset of information, and altogether it contains the information our cells need to survive. But this information is not equally distributed between all 23 chromosomes. Some contain a lot of information, while others very little. And so these are just three examples of features that differ between chromosomes, but we don't understand how these differences might lead to differences in cell division, where specific chromosomes might be affected by an error and possibly lead to a micronucleus formation. And so I really want to understand how these features themselves can inform us on why specific chromosomes end up in micronuclei. So in order to understand how I'll do this, I want to once again have you think of chromosomes as people. So for this example, let's say we have three people inside of a room. Now we can look at these three people and see what features they have similar, that are similar between them. If we look at these three people, it looks like they're all quieter or more introverted, and that's something they all have in common. We can then look at the room they are in, and if this room happens to be a library, we can then try and understand what about their shared features led them to go to the library. In this case, if they're all quieter, maybe they wanted to go to the library to read or maybe to be alone. In this example, we can use the shared traits between these people to inform how they got to their location. So I want to see if we can apply the same thought process to understanding how specific chromosomes end up in a micronucleus. And so how we would do this is that we can identify chromosomes that are contained in a micronucleus and which of the 23 chromosomes are more frequently found in these nuclei. We can then look and see what features these chromosomes share. And then taking these shared features or traits, we can try and understand how that trait led to this chromosome being affected by a cell division error. and then to connect that error to that chromosome continuing on to form a micronucleus. And so how I do this in my lab is we work with human cell culture, where we grow cells in a dish, which gives us a lot of control and allows us to grow a lot of cells at once. So what we can do is take these human cells and treat them with different drugs that will disrupt cell division. And this will cause more micronuclei to form in these cells. So this is an image of these cells. Once again, it's only showing you the DNA in these cells, not the outside of the cells themselves. And you can see each large kind of circle represents a primary nucleus in one cell. And then these small circles around them represent micronuclei. And so these micronuclei can vary in number or in size, but we do, using these drugs, we can get a large number of micronuclei to be produced in human cells. We can then take these cells with micronuclei, and then I break them open. Basically, it's like popping a water balloon where all of the contents are released and we can collect it. We can then take advantage of the fact that micronuclei are so much smaller than primary nuclei and separate out these two groups. And then taking this micronuclei group, I can identify which chromosomes are specifically enriched in these structures. And so I'm still really early on in my degree, and I'm kind of just starting out on this project. So I have a lot more work to do, but I have so far identified 2 chromosomes that we see in micronuclei at much higher frequencies. And so those chromosomes are chromosome 1 and 18, shown here. And so while we still need to kind of do some more work, we can look at these 2 chromosomes and try and see what features they share. For example shown here, both of these chromosomes have a larger than average attachment site. So maybe it's possible that that is what's leading to them going into micronuclei. But it's interesting, why are only these 2 chromosomes affected? There are other chromosomes that have a larger attachment site that we didn't see in micronuclei. And then kind of the ultimate goal of this is to understand how these chromosomal differences and differences in size can actually cause one specific chromosome to be affected by a mitotic error. And I really would like to understand the mechanism of how this occurs later on in my project. And so just to conclude, so far, just from like the few results I have, it looks like chromosome differences can impact cell division, and that we do see specific chromosomes in micronuclei. And so why is this important? This is important because it would allow us to predict errors in which chromosomes would be affected by cell division errors. For micronuclei, these chromosomes will likely have a lot of DNA damage after micronucleus rupture. Also, it would give us a larger understanding of micronuclei's role in cancer. We know they're very prevalent, but we still don't really understand how they're forming or exactly why they're leading to cancer advancement. And with that, I just want to say thank you. Thank you to my lab. It's a really great group of people. To my program in both UW and the Hutch for the opportunity to work on this research. And then also to the Engage program for a really great quarter where we learned a lot. And I learned a lot about communicating science that I'm really excited to apply to the rest of my career. So thank you.
Great. Thanks so much, Molly. I just want to remind everybody that if you do have questions about Molly's presentation, we will do questions for all of our presenters at the end, but feel free to put a question in the Ask a Question field at any time. So I'd like to introduce our next speaker, Andrea Joseph. Andrea is a chemical engineering graduate student at the University of Washington. She studies the brain's natural defense systems and how they go awry in disease. These changes are important to consider when designing new treatments that target the brain. Please join me in welcoming Andrea Joseph.
Hi, everyone. Oh, wow. Almost exactly 24 years ago, I was born 8 weeks early. This is my mom, my dad, and I in the neonatal intensive care unit in Auburn, Alabama, where I spent 4 weeks building up the strength to breathe and to survive without machines. 10% of babies born in America are premature, which I'd bet probably includes some of you listening today. Of course, we're lucky because in the United States, we have the resources to have all these fancy machines keeping babies warm, breathing, and stable. But globally, prematurity accounts for more than 1 million deaths each year, and premature babies are at increased risks for many other diseases as an infant all the way through to adulthood. Because prematurity can be so devastating, there's lots of research being done to prevent it and to treat any following conditions. The Bill and Melinda Gates Foundation have prioritized improving infant health. Closer to home, doctors and researchers at Seattle Children's Hospital created a global alliance focused on this issue. As a fun fact, Anna Faris and Chris Pratt's baby, who you can see here, was born 9 weeks early and received intensive care at Seattle Children's. So now Anna Faris is a board member of this wonderful organization that funds research around the world for premature babies. So from all this research, there have been a few recent advancements in keeping preemies alive, especially in low resource areas. For example, premature babies have underdeveloped lungs. They need to be hooked up to ventilators that can cost $30,000 to $40,000. But recently, Seattle researchers invented a ventilator that can be made from cheaper materials, costing just $1,000 altogether. For infections, which are the leading cause of infant death, global organizations are improving the affordability and availability of antibiotics. For heart, sleep, or weight gain problems, recent research has shown the importance of educating doctors, nurses, and mothers on skin-to-skin care, which can cure many of these issues. But unfortunately, if the baby develops any type of brain injury, if they start seizing or start looking less alert, well, the doctors will try their best treatments, but it's not the same as having a cure. Birth asphyxia is the name of the most common brain condition that occurs in infants, and it's the focus of my PhD research. So birth asphyxia happens when the baby's brain is not getting enough oxygen during some period before, during, or after birth. It can be caused by a variety of conditions, so either a chronic condition that happens during the mother's pregnancy or an acute condition that takes place directly during labor and delivery. As a result of birth asphyxia, babies can go on to develop permanent and long-term brain damage. So survivors of birth asphyxia often develop conditions like cerebral palsy, epilepsy, learning disabilities, or developmental delays that all carry a lifelong burden of disease. Taking a closer look at what's going on in the brain, it probably makes sense to you that a period of time without oxygen is going to be bad for your brain. And this is because if you take a look at the units that make up your brain cells, well, they all need oxygen in order to survive. This oxygen goes towards producing energy, and without energy, cells won't be able to grow and divide, and instead they'll die. But what's really important is the next step. When these cells are dying, they send signals to the rest of your body as part of an inflammatory response. This is called inflammation. Now, thinking about this from a therapeutic perspective, I wanted to know where scientists and doctors could best intervene to stop damage in the brain. Well, these first 3 steps happen pretty quickly. They take between a few minutes to a few hours, and often babies aren't even diagnosed in that time frame. That means our best plan of attack would be to treat the inflammation itself, stop the disease from getting worse. While you might not be familiar with inflammation in the brain, I bet you're familiar with inflammation in other parts of your body. And because of that, you probably have some strategies, some tools in your toolbox to deal with it. If you've consulted your doctor or Dr. Google, you might be thinking of 2 right away: putting an ice pack on it or to take some anti-inflammatory medication like Advil, Tylenol, or aspirin. And actually, both of these strategies have been investigated for the treatment of birth asphyxia. So starting with the ice pack strategy, well, this is actually the standard of care treatment for babies with birth asphyxia. Once babies are diagnosed, they're wrapped in cooling blankets for 3 days. The point of this treatment is to lower the baby's body temperature, which then reduces their metabolism. This means that the cells in their brain need to produce less energy in order to keep living. It reduces the energy demand of your brain cells. That means that we're keeping more brain cells alive, we're having less of them die, and in turn were slowing inflammation. However, this treatment is not effective in severe cases, and it only reduces the risk of death and disability from about 60% to about 45%. This means almost half of all babies with this disease are left with, no effective treatment. This is certainly not a cure. So reaching back into our anti-inflammation toolkit, you might be wondering if anyone's tried anti-inflammatory medication. And while it's not the standard of care, this has been investigated in clinical trials. So here's a clinical trial from 2008 that treated babies with this brain disease with a combination of ibuprofen, which is Advil, and ascorbic acid, which is vitamin C. The results of this study showed that there was no effect of treating babies with these two anti-inflammatory medications. It didn't change the rate of death or the development of brain damage. And actually, it's not surprising that this medication approach didn't work. If we expand our view to all brain diseases, there are quite a few examples that demonstrate how hard it's been to develop drugs that can effectively treat the brain. For example, in the past 50 years, there have been more than 150 failed clinical trials for Alzheimer's disease. There have been more than 50 failed medications for stroke, more than 30 failed treatments for traumatic brain injury, and more than 15 failed drugs for epilepsy. So really, the issue that I'm studying, which is treating babies with birth asphyxia, it's just a window into a larger problem of getting any medication to be effective in the brain. That brings us to our first big question. Why don't medicines work well in the brain? Well, to illustrate this challenge, I have this schematic to show you the journey that we would want a potential drug molecule to take if it would be effective in the brain. You can see that the drug molecule starts here, in a blood vessel inside of the brain. It got here from either being directly injected into your bloodstream, or you took a pill and then the drug got absorbed into your bloodstream. And we want the drug to end up here, inside of the 6 cells of the brain. But this journey is as simple as you might think. In my lab, we like to split it up into 3 segments, each with their own unique challenges. First, we need the drug to get out of the bloodstream into brain tissue. Then the drug molecule needs to navigate the narrow spaces in between brain cells in order to get near the diseased part of the brain. Finally, we want the drug to actually get inside of these sick brain cells. So let's go into a little bit more detail on each segment of this journey so we can then talk about the science of solving these problems. So the first challenge I mentioned was getting the drug molecules out of the blood vessels and into the brain tissue. Here you can see a plastic mold of every blood vessel in an adult human brain. And the first thing you might notice is that there are a ton of blood vessels that feed your brain. Evolutionarily, it makes sense that your brain would need a lot of blood flow because it takes a lot of energy to run the control unit of your body. And it also makes sense that the brain would be really picky about what it takes up from the bloodstream because it shouldn't be vulnerable to infections and other toxic compounds in the same way that the rest of your body can handle. The way that blood vessels can be this picky is by having specialized receptors along blood vessel cells. You can think of these receptors as bouncers because they're standing at the gate between the blood and the brain in control of what gets in and out. So when an essential nutrient comes along in the blood, something that the brain needs, well, these bouncers have no problem letting them into the brain side. But instead, if there are toxic chemicals or infectious materials floating around, well, these won't be let through. Unfortunately, 99.9% of drugs are blocked at this stage too. So that's the first explanation for why a medicine that might work perfectly for the rest of your body would not be effective in your brain. If a drug is able to get past the blood vessels, it then has to navigate these narrow spaces in between brain cells in order to end up in a diseased area. In this microscope image, you can see individual brain cells in gray and the spaces in between them outlined in red. While the spaces in between brain cells together make up about 10 to 20% of the brain, the way that this space is arranged is in really narrow and windy paths, almost like a maze. You can imagine how hard it would be for a drug molecule to get anywhere in this maze. it's likely that the drug wouldn't make it far at all. Instead, it would get stuck or trapped somewhere before reaching a diseased area. So this is the second challenge of getting drugs to work in the brain. They usually get stuck in this maze. At this point, you might be realizing that our one major problem of making effective medicines for the brain should maybe be split into two separate pieces: making effective medicines and then making sure that they can get into and travel within the brain. Maybe the medicines that we already have, like the anti-inflammatory medications or the hundreds of candidates that have been tested for other brain diseases, well, maybe these drugs would work if only we could put them into a delivery vehicle that was optimized for the brain. Well, what would this look like? Let's think about those two challenges separately. The first one was that the blood vessels are really picky about what they let in, and they only let essential molecules through. But what if we could trick those bouncers at the blood vessels by tagging our delivery vehicle with an essential tag, making them think that what we're carrying is an essential molecule for the brain. And the second challenge, the fact that spaces between cells are really easy to get stuck in. What if we coat our delivery vehicle with an anti-stick coating and make it so that the delivery vehicle won't get trapped in the maze and instead would be able to stay inside and travel further? So here I have those two components in a schematic. In the middle in gray is the inside of the delivery vehicle. That's where we can load in any medicine we want and hide it. On the outside, we add our fancy coatings. In yellow, those are the molecules that mimic essential molecules to trick the blood vessel bouncers into letting us into the brain. In black, we have an anti-stick coating to prevent getting trapped in the brain cell maze. So what we came up with together is this amazing nanoparticle. It's a nifty new technology that my lab is studying because we think they can solve the current problems with developing effective treatments for the brain. They're called nanoparticles because they're nano-sized. They're very small. If you think about the height of one dime, you'd need a sack of 10,000 nanoparticles to reach that height. Their small size and the special surface that we designed make nanoparticles really promising for better drug delivery in the brain. So let me give you a few pieces of evidence that these nanoparticles really do target the brain. Our first prediction is that those yellow essential molecule tags on the outside of these nanoparticles really do allow them to get across blood vessels better. To test this hypothesis, I injected both types of nanoparticles, the tagged and non-tagged, into healthy rats and measured the amount of nanoparticles that got across blood vessels into the brain. And what you can see here is that tagging did result in a 5-fold increase in the amount of nanoparticles that were able to get into brain tissue. So the tagging really did work. Our second prediction is that anti-stick coating would allow nanoparticles to travel through that brain cell maze, to travel farther in the brain compared to nanoparticles without that coating. This is an experiment that my PhD advisor, Dr. Elizabeth Nance, did during her PhD, where she directly injected both types of nanoparticles directly into a rat brain. 1 hour later, she took a microscope image of how far both nanoparticles traveled. And you can clearly see that those green anti-stick coated nanoparticles did travel farther in the brain compared to the orange ones without that coating. Okay, so I've given you some proof that the nanoparticle technology really does overcome those first two challenges in getting molecules inside of the brain. It helps molecules get across blood vessels and navigate those narrow spaces in between cells. But what I haven't yet shown you is how we expect our nanoparticles to target, to identify, and get inside of just sick brain cells. And it's really important that we do target the sick and not healthy cells, because when you start getting drugs to healthy areas, that's when side effects or toxicity could happen. From this picture, you can see why that could be challenging. There are so many different types of brain cells that are involved in many different diseases. And these cells are so interconnected and overlapping. What we need is a way to identify which cells are sick and then get our nanoparticles inside of them. One solution to this problem is to be strategic about the type of cell that we're targeting. Sick immune cells in specific turn out to be a great target for nanoparticles because their job is to eat up foreign stuff. I really like this video because it looks like the scientist is having a lot of fun playing around with the immune cell, trying to confuse it on where the foreign object is. But it's also really cool to see how the sick immune cell naturally knows from chemical cues which way to move in order to eventually eat up this particle. So we were confident that in the same way, sick immune cells would be able to also eat up our nanoparticles. So I did this experiment where I applied my nanoparticles to both healthy and sick brain immune cells. And you can see that once again, our hypothesis was correct, that nanoparticles did accumulate in the sick immune cells 5-fold more than they did in the healthy ones. Okay, so at this point, you now have an idea of the challenges involved in getting drugs to work in this brain and how we designed our nanoparticles to overcome these challenges. I want to wrap up by showing you how we applied this brain-targeted nanoparticle technology for the development of a new treatment for birth asphyxia. Well, we had our delivery vehicle, the nanoparticle. We just needed an anti-inflammatory drug to put inside of it. And what if I told you that there was a really trendy anti-inflammatory drug sitting in your kitchen cabinet right now? This is curcumin. It's the active ingredient in the spice turmeric, which is used a lot in Indian cooking. And many studies have shown that it's really good at reducing inflammation. It's because of this, it's been investigated for the treatment of many diseases, including arthritis, anxiety, and exercise-induced inflammation. But unfortunately, it can't accumulate in the brain, just like 99.9% of drugs. That's okay because we thought our nanoparticle could help curcumin overcome that challenge. So that's me in lab on the left, making the materials for these nanoparticles. And on the right, you can see how the nanoparticles turned out. In order to test if these nanoparticles would be effective at treating birth asphyxia brain injury, we had to compare treating rats with that brain injury with the nanoparticles in comparison to rats treated with the curcumin on its own. But once again, we expected that that would be unsuccessful since the curcumin could not actually get into the brain. So when we had these birth asphyxia brain injured rats and gave them no treatment, you can see that most of the brains ended up with really severe brain injury. After treating them with just curcumin, you can see that that didn't really do much. Still, most of the brains looked like they had really severe injury. However, after treating these animals with curcumin nanoparticles, we totally flipped the distribution of injury that we saw. In fact, a significant fraction of the animals in this group actually looked like they had totally healthy brains. So this was a really exciting and significant result for us. It validated our idea that we can create more effective medicines to the brain by putting promising drugs inside of delivery vehicles that are optimized for targeting the brain. So that's the idea I've been studying for the past 4 years in my PhD research, and hope to continue studying, ultimately with the goal of curing brain disease in newborns for good. Thank you to my wonderful lab and our collaborators who have allowed me to do this exciting research. Thank you.
Great. Thanks so much, Andrea. Again, if you have a question, feel free to put it in the question box at any time. And now I'm going to introduce our final speaker, Laura Carlucci. Laura is a grad student in the Department of Bioengineering at the University of Washington. She is studying the effects of mechanical forces on molecular binding, as well as the mechanism behind a particularly force-resistant interaction found in nature. Please join me in welcoming Laura Carlucci.
Sorry, slight issue with just getting my PowerPoint up. Okay. So thank you for that introduction, and I'm really excited to talk to you all about my research. And the title of my talk is Don't Just Go with the Flow. how some bacteria use pulling forces to initiate infections. So, you know that feeling when you really gotta pee? That pressure in your lower belly and how the only thing you can think about is where's the nearest toilet. But then once you do make it to the bathroom and relieve yourself, you feel much better. Unfortunately, for the many individuals living with a urinary tract infection, they always feel this need to have to pee. And if they do make it to the bathroom and relieve themselves, it's not exactly a painless process. Unfortunately, urinary tract infections are incredibly common, affecting up to 150 million individuals each year. And this number is made up of primarily biological females, just because their anatomy of their urinary tract makes them more susceptible to these urinary tract infections. In fact, up to 60% of all women are expected to develop a UTI within their life at some point. And then once they develop a UTI, and then get over it, the chances of getting a reoccurring infection are rather high. And in fact, these infections are so common that they make up 25% of all total bacterial infections. So since these are so common, wouldn't it be great if we can develop some sort of drug or preventative therapeutic that we can take that will minimize the frequency of these UTIs? Well, before we can develop such a treatment, we first need to explore what causes these infections. So as I mentioned, UTIs are a form of bacterial infection, which means they're caused by bacteria. And bacteria are made up of cells, kind of like how we're also made up of cells. And Molly introduced you a little bit to cells and about how they're basically just little packages that contain stuff like DNA and the little structures that the cells use for them to do their job. And our cells work together to make, well, us, whereas bacterial cells tend to behave a little more independently from each other. And so a UTI occurs when one of these bacteria somehow gets inside of our body, say through our urethra, which is the tube that connects our urinary tract to the outside world. So the bacteria can travel up to our bladder, and if it makes its way all the way to our kidney, it can cause some really severe complications. But luckily, our body has a natural defense mechanism against these invading bacteria. It's urinating. So when you go to the bathroom, urine is flowing out of your body, and this generates forces that are literally washing the bacteria away. However, not all bacteria are washed away during this, and unfortunately, these bacteria are often associated with urinary tract infections. So what is it about these bacteria that lets them hold on to the walls of our urinary tract? Why is it that only these bacteria are so strong that they can resist the flow of urine? Well, it turns out in the absence of any flow or forces trying to wash the bacteria away, these bacteria aren't really remarkable. They're just swimming around and they may grab onto the walls of your urinary tract, but they can't get a good grip. They let go very, very quickly. But when there's a flow of urine and there are now forces trying to wash the bacteria away, these bacteria actually become strengthened by the forces that are trying to wash them away. And so as a result, as urine is flowing past the bacteria, they can now grab onto the walls of your urinary tract and hold on tightly so they can now stick around. So what is it about these bacteria that let them do this, that are suddenly become stronger with force? Well, they have a secret weapon. On the surface, they have these little hair-like appendages that contains a molecule called FIMH. And FIMH is what's known as an adhesive molecule. And adhesive molecules do what you can probably guess they do. They stick to things. And of course, FIMH will stick to the walls of your urinary tract. But in particular, it likes to grab onto this molecule called mannose, which is just naturally found on the surface of your urinary tract. So FimH grabs onto mannose, and as a result, the bacteria are able to stick to the walls of your urinary tract and resist the flow of urine that's trying to wash them away. But as I mentioned before, this only happens when there is actually a flow of urine. Otherwise, in the absence of any forces trying to wash the bacteria away, the bacteria are able to freely move around and don't really stick to anything. So why isn't it that FimH isn't grabbing onto mannose all the time? Well, it turns out the secret is the force that's trying to pull the FimH molecules away from the mannose molecules. When there's such a force, FimH is literally changing shape in such a way that it helps it now hold onto mannose tighter. And now with this tighter grip, it is able to remain attached to the walls of urinary tract, and as a result, the bacteria don't get washed away. Okay, so this is a pretty unintuitive concept, but you might have actually seen something like this if you've ever played with one of these toys. This is a finger trap, and they work by putting a finger of either hand in either end of the toy. When you move your hands apart, the toy literally tightens around your fingers, trapping your fingers together, and so you can't get your hands free. And in fact, the harder you pull your hands apart, the stronger the toy holds onto your fingers. And to eventually get your hands free of the toy, the secret is to just slowly pull your hands apart, essentially separating them with very little force, so that now the toy can't tighten around your fingers, and thus you can slip your fingers free of the toy. And so FimH works in a very similar way. When you try to pull FimH away from mannose, FimH changes shape in such a way that it now tightens around mannose, thus giving it a much better grip. And so it's a lot harder for FimH to eventually let go of mannose. We don't really see the two molecules coming apart when we pull on them in this way. And so FimH's shock absorbing property essentially just comes down to the fact that it has these two distinct shapes. One that holds onto mannose weaker, and another that holds onto mannose stronger. And FimH can transition to the stronger shape when there's a force trying to pull it away from mannose. And so we can conclude that in the urinary tract, when there is a flow of urine, there are now forces trying to pull FimH away from mannose, and as a result, FimH is going into the stronger shape. In the stronger shape, this helps the bacteria stick to the surface of your urinary tract and not be washed away. And so as a result, this second stronger shape is playing a very important role in helping these bacteria infect your body and cause a urinary tract infection. And so therefore, maybe we want to create some sort of pill or drug that will bind to FimH when it's in the shape and mess with its ability to hold on to mannose. If FimH can't get a good grip on mannose, then the bacteria can't stick to the walls of your urinary tract, and this will just be washed away when you use the bathroom. But before we can start developing such a preventative drug that does this, we need to be sure that FimH just has these two shapes, that there isn't some additional third shape that we just don't know anything about. Because if it turns out that FimH can change shape once it's in a stronger shape to another additional shape, that would really complicate our ability to design such a drug that helps protect our bodies. So in a sense, we want to investigate whether or not this third shape can possibly exist. Now, unfortunately, determining the shapes of molecules isn't easy. We can't exactly just look at the molecules. So instead, we have to try to study, examine some other property or behavior of these molecules, one that we already know how to measure. And such a behavior that we do know how to measure is how strong FimH acts when you pull on it. So to explore this idea, let's go back to the finger trap. As I mentioned before, if you have your fingers in either end of the toy, the harder you pull your fingers apart, with more force you pull them apart with, the stronger the toy behaves and traps your fingers with. But let's say you are really strong. So strong, in fact, that you pull so hard that the toy cracks. Now from here, if you were to keep pulling, eventually the toy will break in half and you'll get your hands apart. And so we would say, in a sense, that with additional high force, the toy is weakening. So let's bring this back to FimH. We know already that if we pull on FimH, it changes shape, and which gives it a tighter grip to hold on to mannose with. But if we keep pulling with enough force, eventually the two molecules will come apart. And with that, we can get an idea of how strong FimH is behaving as we're pulling on it. And so we can look for patterns on how strongly FimH is holding on to mannose under different forces to draw conclusions about the shape of FimH. So let me explain this. So let's say we take FimH and we pull on it, overcoming its shock absorbing ability, so now it's in its strong state. And then we just keep pulling. Well, if we pull on it with relatively less force, FimH has a pretty good grip on mannose. It'll be able to remain holding on to mannose for quite a while, and it's going to be really hard for the two molecules to come apart. They'll come apart pretty slowly. But let's say we do the same thing, but then we keep pulling on FimH with a lot of force. Well, if that's the case, we would expect FimH to let go of mannose pretty quickly, because after all, it's being pulled on with a lot of force. But let's say, alternatively, FimH can change shape. Once it gets into strong shape, it has the ability to change into another shape. So if we pull on it with a little force, this may cause it to change shape, and we may not see it let go of mannose, but this will become even more apparent when we pull on it with even more force, and we still may not see it letting go of mannose. And it may just take a lot more force or a lot more time before we eventually see the two molecules coming apart. So we can take this idea of taking FimH and pulling on it so it gets into a strong state, and then just keep pulling and see, how does FimH behave? How strong is FimH when we keep pulling on it? And so based on how it behaves, the strength of FimH, we can draw some conclusions about the shapes that FimH is taking on. Let's say, for example, FimH doesn't change shape. Once it gets into its strong shape, it stays in its strong shape. So then we would expect that the strength of FimH would decrease as we pull on it with more and more force. And it would decrease in such a way that it forms this straight decreasing line. But let's say it turns out that FimH is changing shape. Well, then it's a little bit harder to predict how the strength of FimH is going to change. Most likely it will possibly increase before eventually decreasing, or some possible two-direction lines. So now we can translate these ideas into an experiment that we can do. So we take FimH and we pull on it so it gets into its strong shape. And then from there, we just keep pulling on it at many different forces. And then we just measure how strong is FimH behaving as we're pulling on it with all of these different forces, and look at the shape of the resulting graph. Based on the shape, we can draw some conclusions about what FimH is doing. Now, the next question we need to address is, how do we pull on molecules? Well, in my lab, we have an instrument called a magnetic tweezers. And a magnetic tweezers is just a microscope with a magnet on it. And the microscope is looking inside of a chamber, and glued to the floor of the chamber are FimH molecules. And then inside are magnetic beads that are free to move around. And then we add mannose molecules around these magnetic beads and place magnets above and below the chamber. So we can use these magnets to move the magnetic beads up and down, and say use the bond magnets to pull the beads down. And now FimH can grab onto any mannose molecules that it can reach. And then we use the top magnets to pull on these magnetic beads and essentially pull on FimH as it's holding on to mannose. And we can control these magnets in such a way that we pull on it with a certain force that we choose to apply. And then to measure how strong is FimH as we're pulling on it, we just time and see how long it takes until FimH eventually lets go of mannose and the magnetic bead flies away. So I measured this for many different molecules of FimH with many different pulling forces, and I measured how strong FimH is acting under all of these different forces. And I'm going to show this in a graph, but just as a refresher, what we're interested in is the resulting shape of the graph. If it turns out the graph forms a straight decreasing line, that would suggest to us that FimH isn't changing shape. once it gets into strong shape, it stays in the strong shape. But if the line is a little more complicated, possibly increasing before decreasing, then that would suggest to us that FIMH is changing shape. So I'm going to show my data here on this graph, where on the horizontal axis I have the force that I was pulling on FIMH with, and the vertical axis is just how long FIMH was able to hold on to mannose, which correlates with how strong FIMH was behaving. So here's my data, and let's take a look at it. Does it look like a straight decreasing line or something else? Well, as I look at it, it looks rather like a straight decreasing line, and I'm able to fit a line through the data fairly well, which suggests to me that once FIMH gets into its strong shape, it is staying in this strong shape. And so now we have a little more confidence in how FIMH is actually behaving. It has this weaker state that when there's some force trying to pull mannose away from it, it now transitions to a stronger state. And this makes sense in the context of our urinary tract. In the absence of any flow of pee, there are no forces pulling on FIMH, and so the bacteria don't really have a good grip on the walls of our urinary tract. But then once we go to the bathroom, this generates forces trying to wash the bacteria away. This causes FIMH to transition to its stronger shape, and so the bacteria can stick tightly to the surface of the urinary tract. But then once this flow of urine stops, the force disappears, and now FIMH reverts back to its weaker shape. From here, it can't hold on to the walls really well, so the bacteria are free to let go and continue moving on and causing urinary tract infections. And so now that we have a better idea of how these bacteria are getting past our body's natural defense mechanism and invading our bodies to cause urinary tract infections, we can try and design some sort of pill or drug that will help protect our bodies against these invading bacteria and decrease the frequency of urinary tract infections. So I'd of course like to thank everyone in my lab and Wendy Thomas for being a great mentor, the Engage program at UW for putting on the class, and the instructors for giving excellent feedback, and Town Hall Seattle for providing this platform for me to talk to you all about my research. Okay.
Cool. Thanks so much, Laura. I liked your Ren and Stimpy image there. That's really great.
Yeah, it drives home the point.
Yeah, definitely. Okay, so we're going to go through questions now. Again, if anyone has a question, just throw it into that Ask a Question field. And we're going to start with Molly. So I'm wondering, Molly, can you kind of explain what stimulates or what's the catalyst for a cell to begin to divide?
Yeah, so it differs depending on the cell you're talking about. During development, this just happens very rapidly all at once. But then there's different places in our body where cells divide kind of at different times. Our skin cells are constantly dividing because they have to kind of replenish themselves. But there's other places where kind of signals can be sent to a cell to tell them to divide. So it can kind of differ depending on what region of the body you're looking at, I guess.
Okay. All right. And then this question comes from Dennis. He is asking, could 1 and 18 chromosomes be affected because 18 has a large attachment site and that can result in poor attachment, and 1 because it's so large that it, I think he's saying that it drags while it pulls towards one side, so it's heavier on one side maybe than it is on the other. Yeah.
So those are both great. That's kind of the next step, trying to identify what's causing this. For 18, we know from other groups that a larger attachment site doesn't lead to poor attachment. It actually leads to more attachment. multiple kind of ropes can attach to one chromosome. And the issue is that when you can form more attachments, you can form more incorrect attachments, where one chromosome can be bound by ropes kind of coming from both sides. So then when they're trying to pull apart, this one chromosome is being pulled in both directions. So that's one kind of mechanism where the attachment site can cause an issue. And then for chromosome size, it's been shown that larger chromosomes don't actually have more drag. There's been some kind of biophysical studies on that I don't know all the details of, but they've shown that chromosome size doesn't affect in that way, but we don't know. It seems like it is important, but we just don't know the exact mechanism that size is actually leading to these differences.
I think it's really interesting to think of things happening in the body as like almost mechanical, and like you talked about a rope, like that is really helpful to understanding it.
Yeah.
This next question is from Jan. Are cancer cells and rapidly dividing cells more prone to forming micronuclei?
Yeah. So it's really, cancer cells are especially prone. Even the cells that we use in lab, we study cells that are obtained kind of from epithelial cells, so like skin cells, and they divide very quickly, but they don't form micronuclei actually. We have to kind of knock out, like disrupt something or treat them with a drug to make them form micronuclei. So we really see these mostly in cancer cells, where cancer cells just have a lot of mutations and a lot of things going wrong that led them to become cancerous. So because of all those existing issues, they just form more errors in cell division to begin with.
And then once you're sort of able to understand a bit more about what is in micronuclei, what can be done with that information? What could be changed to prevent that from happening, or is that still a little unclear?
Yeah, so I think the main thing is it would be really great to have an understanding of kind of how specific chromosomes are affected. We also see in certain cancers, specific chromosomes are either gained or lost in specific tissues. So it looks like there are chromosome-specific effects, but knowing how this affects micronuclei, we'll know which chromosomes are likely to be damaged or kind of shattered when the micronucleus ruptures. And also we can kind of take the knowledge and work backwards, where if we see specific chromosomes in the wrong cell or in a micronucleus, we can then kind of know what cell division error might have led to that, to have an understanding of what mutations exist within that specific cancer cell. So for specific treatments, it might be kind of a little more in the future. My research is more basic science, understanding kind of like how cells work to later inform treatments. But really just understanding how chromosome differences in general important is what I'm really excited about, because we really just don't have a good understanding of that in the field right now.
All right. Great. Thanks so much, Molly.
Thank you.
Really well done. So, Andrea, we're going to move to you next. Your sort of beginning talk about your own experience was really, really interesting to hear. I heard a radio program recently about how helping keep preemies alive. Some of the way that incubators came about was like traveling around in carnivals, and like it's very strange. It was a very strange history of that, but I think it's really great that you've come to this through your own personal experience. And my wife is also a preemie, so it's pretty interesting.
Wow.
Happens a lot. But I am wondering if you can talk a little bit about how a drug usually is able to get to the part of the body that needs it. Like, how does a drug, when you just take an aspirin, find its way to, say, your knee that you have, fallen and tripped and hurt yourself? How does it get there typically?
Yeah, that's a great question, because we often feel like, my knee hurts and I take aspirin and it immediately works, or I feel like my knee is getting better. But what's important to recognize is that there is no specific targeting ability of those chemicals. So really what you can imagine is that these drugs are just circulating throughout your whole body pretty much equally. There is a slight effect, in areas that are inflamed. They might be receiving an increased blood flow because that's part of how your immune system responds to, that's part of what inflammation is, right? Swelling and a temperature increase and things like that. That is part of the natural way that your body can direct nutrients and resources that are flowing around in your circulation to these areas of injury. But there's nothing that's comparable to the way that we're targeting nanoparticles for specific organs based on like the identity of brain cells compared to knee cells or something like that. So it's a different level of specificity compared to drugs that you would recognize like on the market today.
Okay. And so the drugs have a hard time getting to the parts of the brain that need it. Mm-hmm. And so once they're in the body, though, and they have stopped at whatever point they're able to get to, what is the body able to, are there side effects because it hasn't gotten to where it's supposed to be in the brain?
Yeah. So when I'm thinking about our anti-inflammatory drugs, there really shouldn't be any toxicity associated with delivering anti-inflammatory compounds to an area that doesn't exactly need it. This is probably similar to just taking a multivitamin to keep everything healthy at a normal level. There can be, at an extreme example, if you're thinking about delivering like chemotherapeutics or any drug that's supposed to like kill wherever it ends up, that would be an extreme example of where you really need to make sure that you are successfully targeting sick areas if you wanted optimal like delivery efficiency, which I understand is not our current situation. But that would explain, I think, why even if there are side effects from delivering anti-inflammatory drugs to areas that aren't diseased in the brain, you aren't seeing any negative results because of that.
Okay. And I've got a question here from Taryn, who's asking, Andrea, could you tell us more about why curcumin specifically is a more effective anti-inflammatory than others for birth asphyxia?
Sure. Thank you, Taryn. Taryn was the instructor for the Engage course, and I think we were all really fascinated by the power of curcumin, how I focused on that in my research. But actually, the story of why we picked curcumin isn't too special. I mean, as I mentioned in my talk, it's pretty trendy. So there's a lot of recent studies that have focused on curcumin. But in the beginning, our criteria wasn't that it wasn't that specific. We were interested in any anti-inflammatory compound that had positive effects in brain cells. So there are some things that curcumin is great at promoting, specifically the health of neurons in your, so the specific communicating cells in your brain. But the most important reason why we chose curcumin was because it was anti-inflammatory and also an antioxidant, which is true of many different compounds.
And then you mentioned other brain diseases that have been resistant to drug treatments. Is it possible that this research that you're doing will be, if it is successful, would be able to help those as well, or are those delivered differently?
That is absolutely our research hope, goal, and dream. So there currently isn't a cure for any brain disease. While I focus on birth asphyxia in my research, my lab mates do research in models of autism, depression. We are collaborating with a group who does research with Alzheimer's, a model of Alzheimer's disease. And so the hope is that nanoparticles that are able to target the brain would be able to target the brain across the variety of diseases. And drugs that we can put in there that are loaded, that are anti-inflammatory, would be able to treat inflammation, which is relevant for many and nearly all brain diseases.
Okay, great. Well, thank you so much, Andrea.
Thank you.
And finally, Laura, can you talk a little bit about how drugs that we would get, this is actually kind of a, now that I think about it, kind of a similar question to what I just asked Andrea, but can you talk a little bit about how drugs work, like over-the-counter drugs that are typically purchased to treat a UTI, like how those work and how that interacts with your research, or how it's maybe how those are just not good enough.
So I actually know of one preventative drug that is available over the counter for people who take to prevent UTIs, because there are certain instances when you have a higher chance of contracting a UTI, like some people just have a higher chance, just like biologically, and also after intercourse, females have a higher chance of contracting it. So I know there's like over-the-counter preventatives currently available, and one of them actually behaves like mannose, or is mannose, which is the molecule, if you recall, that FIMH likes to bind to. And so the idea is if you flood the system with mannose, mannose will essentially like clog up the FIMH molecules, and so they can't bind to your urinary tract, and thus they'll get washed away, similar to what I was talking about our like proposed possible drug. It's just it's imperfect because, as I mentioned, in the absence of like a force, FIMH doesn't hold on to mannose super well. So it works, but it could be better.
Okay, interesting. And the imagery, or the instance of something attaching more firmly when pulled from, like, do you know if there are other instances of this happening in the body, like other just other examples?
Yeah, so that's actually an incredibly great question, because right now the field of molecules that get stronger, that hold tighter when you pull on them, it's currently an emerging field. Like the first instance was found maybe like 20 years ago or so, and since then we've been discovering more and more molecules that do this. And some of the most well-studied ones are actually the molecules that hold our cells together, since so our cells, they can essentially communicate with each other through like the molecules that are holding them together. And one way they do that is by sort of a pushing and pulling sensation. So kind of like how I might poke a friend to like get their attention, or like pull on like their arm. Cells actually work in a very similar mechanism, but how it works for those cells, it's a lot more complicated, because there's actually like multiple molecules involved, and it forms like a chain reaction. And so there's a lot of research being done on it. And one motivating aspect of my research outside of the UTI aspect is understanding how FIMH works, because this property is so interesting and is found in more and more molecules. Like every year, we're discovering more and more molecules that have this behavior. But FIMH is actually relatively simple to study, as far as, since it works basically on its own rather than having like in a complex of other molecules.
And can I ask what got you into this particular study? How did you arrive at studying this?
Yeah, so one of the things that just drew me to science altogether was just how stuff like chemistry and physics and stuff like that, just how are we made up of molecules? Because in a sense, everything is made up of molecules, but how can I be like a thinking creature that is made up of molecules? So that's like the big broad philosophical thing that got me to science. And then it was actually my mentor who, she was working on this project, and so she brought me onto it. And I was just fascinated by the idea of how like literal like physics, like pulling forces that we think about in terms of just like the big world scale of like, if I pull on a finger trap, like we think of it on like the big scale of how that applies to molecules, and how molecules are actually employing the same properties, the same things that we study on like the big scale, how they're still applicable at like the small molecular scale, and that a lot of biology is designed around, like has evolved around that, these properties.
Great. Cool. Well, thank you so much, Laura.
Yeah.
And thank you to all of you for presenting today. I have to say this is the last one and of this series. I've been really inspired by all the students and all the really interesting work you're doing for things that are currently issues that need to be solved, and for even moving into the future, things that we haven't even thought of yet. So thank you all for all the work that you're doing and for presenting with us tonight. And thank you all for watching. If you are interested in more Town Hall content, you can follow this Crowdcast channel by clicking on the follow button at the top right corner of the page. And we will see you next time.